Okay, so here we are with chapter 19, chromosomes and cell division. We'll look at the two types of cell division and we'll follow along this outline. So again, it goes along nicely and it builds off of reproduction. Now we'll kind of look at cellular reproduction. So we begin life as a single cell or a zygote formed by that fertilization or union of an egg and a sperm. And the human life cycle has two types of cellular division. Meiosis is the nuclear division that gives rise to gametes, or the sex cells, and mitosis is the nuclear division that results in identical body cells. So most of our cells go through mitosis, but our sex cells, our eggs and sperm, go through meiosis. I'll have you guys watch this video on your own. Meiosis gives rise to the gametes that have half the number of chromosomes as the original cell. And that's because in females with our ovaries producing eggs and in males with the testes producing sperm, each egg and each sperm has half the number of chromosomes. So with that, when they unify or join during fertilization, they'll make a complete set. Mitosis results in identical body cells, and this occurs during normal growth and repair of pretty much all your cells. So here's a look at how meiosis is formed and where it produces gametes. The male gamete is the sperm, the female gamete is the egg, and upon fertilization, we call them 2N. So we say it's N, and that just represents one set of chromosomes or haploid. And then if they come together, we call it 2N or diploid, which is two sets of chromosomes. Again, one set of chromosomes is 23, two sets is 46. 23 plus 23 is 46. And then here we have a baby, and then mitosis will produce cells for growth and repair throughout someone's life. Chromosomes are located in the nucleus. Each is a tightly coiled combination of DNA and specialized proteins called histones. The DNA contains genetic information which directs body development and maintenance, and the histones just help with support and control of gene activity. A gene itself is a specific sequence or segment of DNA, and this will direct synthesis of a particular protein, which will play a structural or a functional role in the cell. Somatic cells, soma is body. These are all your cells except eggs and sperm, which we call gametes. Humans have 46 chromosomes, two sets or two pairs of all 23. One set of 23 you get from each parent. And each somatic cell contains two chromosomes with genes for the same traits. They're called homologous pairs, where one chromosome of the pair is from the mother and one chromosome of the pair is from the father. Diploid refers to a cell with two of each chromosome, or 2N, and genes also occur in pairs in diploid cells. So members of each gene pair would be located on the same position on the homologous chromosome. Of the 23 pairs of chromosomes, the sex chromosomes make up one pair, and they'll determine the gender. XX is a female, XY is male, and autosomes make up the other 22, and these will determine the expression of most of a person's other inherited characteristics. So mitosis is the process of cellular division. The cell cycle itself is the predictable set of events that a cell goes through from its origin to its own division into two daughter cells. There's two major phases of the cell cycle, interphase and cell division. And if we look at interphase, this is where normally a cell spends most of its time. An interphase just includes a growth, synthesizing DNA, and more growth, and then a more growth stage. The cellular division stage is where we get the different steps of mitosis from prophase, metaphase, anaphase, telophase, and then cytokinesis is the actual splitting of the cell. So interphase is the period of growth and preparation for cellular division. It's not necessarily a resting period because there are three distinct phases in it, and a lot of growth happens during these phases. I'll let you guys read through the G1 phase of interphase, the S phase where DNA is replicated, and then the G2 phase. So here's a look at the cell moving towards interphase, how one duplicated chromosomes involves two chromatids, and then they'll eventually condense around and wrap around histones. And then the DNA will eventually twist and loop around itself to create its sister chromatid. So one chromatid is here, the other sister chromatid is here, and they meet together in the center at the centromere. The division of the nucleus and cytoplasm occurs in mitosis, which divides the nucleus, and then cytokinesis is what divides the cytoplasm. These are the stages of mitosis. DNA replication during interphase forms two sister chromatids, which are banded together to form a chromosome. During mitosis, the sister chromatids separate and move to opposite sides of the cell. And then during cytokinesis, the parent cell will divide into two copies of each chromosome, or homologous pairs. The division of the nucleus and the cytoplasm, it happens during the development of the embryo and fetus, where the body cells are dividing continually. It also happens in children for the growth and repair. However, when you get to adulthood, some of the cells lose their ability to divide, like your neurons. Some cells stop dividing, but retain that ability, like your liver cells, but in some cells actively continue dividing throughout your life. So these are the four phases of mitosis. I always think of the acronym PMAT. So prophase, metaphase, anaphase, telophase. PMAT, to remember the order. And I'm going to let you guys kind of look at the order and what happens at each phase, but you should know what exactly is happening at prophase, metaphase, anaphase, and telophase. And this should be a video that takes you through those four stages of mitosis that you should know well. And simply put, here are some pictures that I'll go through. In the interphase, the DNA is duplicated, growing. Prophase, the chromosomes condense. Metaphase, the chromosomes will line up across what we call the equatorial plate in a nice line. And the pictures above are actually cellular pictures, which is kind of fun to see a cell in real time going through these stages. Anaphase, the sister chromatids are pulled apart. Telophase, we start to see what we call a cleavage furrow or an indentation break apart the cells. And then cytokinesis is actually the complete breaking apart of two daughter cells. Cytokinesis is the division of the cytoplasm. It will begin during telophase, eventually pinching the cell in two. And this is just a look at cytokinesis occurring right after telophase. Karyotypes are constructed by arranging the chromosomes from photographs, and they can be examined for defects in the number and structure of chromosomes. So this is a karyotype showing all 23 pairs. And you'll see all of the up to 22 are your autosomes, and the 23rd pair is the sex chromosome, the XY. So this would be a male set of chromosomes. So the karyotype you can look at for defects and zoom in on a particular gene sequence to see if anything could possibly arise for future problems. That individual. So gametes are eggs or sperm, and again, they differ from somatic cells. So this is our introduction into meiosis now. Gametes are haploid, and we refer to that by the letter N, and that just means they have only one member of each homologous pair of chromosomes. So there's 23 chromosomes. They're formed by meiosis, which involves two divisions that result in up to 4 haploid daughter cells. And the functions of meiosis are to keep the number of chromosomes in a body cell constant from generation to generation, and this will increase genetic variability in a population. So meiosis involves two cellular divisions. Meiosis 1 will separate homologous pairs of chromosomes, and meiosis 2 separates sister chromatids. Each division has its own PMAT phase, the prophase, metaphase, anaphase, and telophase, and they're very similar to the PMAT phases in mitosis. So this is showing the diploid parent cell contains two homologous pairs. DNA replication occurs, and during meiosis 1, the homologous pairs separate, and during meiosis 2, the sister chromatids will separate, so we get 4 haploid daughter cells. So you'll notice from meiosis 1 to meiosis 2, we're just separating out the sister chromatids from the chromosomes to get haploid gamete cells. So here is meiosis 1. I'm going to let you read through this on your own. Prophase 1, metaphase 1, anaphase 1, telophase 1. So again, meiosis has these 2 cellular division phases, and they both go through the PMAT phases. Interkinesis is the brief interphase-like period before meiosis 2. And then we get into meiosis 2, and we go through the prophase 2, metaphase 2, anaphase 2, telophase 2. Cytokinesis will occur at the end of telophase 2, forming those 4 haploid daughter cells. And again, the important feature of meiosis 2 is that it separates the 2 sister chromatids of each chromosome. So this is a great video of meiosis that I would also encourage you to watch. And this kind of briefly goes through meiosis 1 and meiosis 2, resulting in 4 haploid daughter cells. So again, this is showing a parent cell that is diploid or has both sets of chromosomes, and whether it goes through mitosis or meiosis. If it goes through mitosis, it'll create 2 identical diploid cells, the 2N, and if it goes through meiosis, it will create 4 haploid cells. Following meiosis, changes in the shape and function of the haploid cells result in functional gametes. So in spermatogenesis, which produces sperm cells, it will produce 4 sperm cells specialized for transporting the male's genetic information to the egg. But in oogenesis, it produces up to 3 what we call polar bodies and only 1 ovum packed with nutrients to nourish the early embryo. So in spermatogenesis, you get 4 sperm, but in oogenesis, you just get 1 egg and 3 polar bodies out of it. So here's a look between sperm formation and egg formation. During meiosis, genetic variation is created through what we call crossing over, and this happens when corresponding pieces of chromatids of the maternal and paternal homologous chromosomes are exchanged during synapsis in prophase 1, and also independent assortment, which is the relative positioning of homologous maternal and paternal chromosomes with respect to the poles of the cell. So let's take a look. This is showing how crossing over occurs between non-sister chromatids, and you can see where crossing over occurred with a specific gene sequence or portion of that chromosome, and then it'll result in chromosomes that have a new combination of maternal and paternal forms of genes. So this is showing 2 equally possible ways that homologs could align during metaphase 1, and then showing in metaphase 2, and then how you would get different possible combinations depending on how they will align during metaphase 2. So here's a look at comparisons between mitosis and meiosis and kind of the differences between the 2. You should know the differences. The big thing is that during mitosis, you produce daughter cells that are genetically similar. And again, mitosis occurs with most of the cells in your body, whereas meiosis has 2 cellular divisions. It produces the haploid egg and sperm cells, and these daughter cells will be genetically different. Non-disjunction, excuse me, is the failure of homologous chromosomes to separate completely, and non-disjunction could result in too many or too few chromosomes in a cell. And this could cause some sort of abnormalities in development and miscarriage. And this is just an example of non-disjunction, how it shows you missing 1 chromosome, or if you have 1 additional or 1 missing, and it would usually result in the spontaneous, non-functioning of the cell. Trisomy is 3 representatives of a chromosome in a cell, and an example is Down syndrome, where an infant is born with 3 copies of chromosome 21, The risk of having a baby with Down syndrome usually increases with the mother's age, and this could cause multiple physical and mental abnormalities. Monosomy is only one representative of a chromosome in a cell, and non-disjunction can also occur with sex chromosomes, where the non-disjunction of a sex chromosome during the sperm formation could result in a sperm that carry both X and Y, or no sex chromosome at all, and non-disjunction of a sex chromosome during egg formation, the resulting egg will have two X chromosomes, or none at all. And examples of this are what we call Turner syndrome or Klinefelter syndrome. So this is showing Turner syndrome or Klinefelter syndrome in females and males. From this, you should be able to go through these slides, and that kind of ends our reproductive section. We have one more chapter to get through before this week 4 exam or module 4 exam. So thanks for listening.